Hello everyone, welcome to Agri Clarity, your go-to channel for agricultural knowledge. Today we are diving into a crucial topic which is biofortified versus bioengineered, which is genetically modified crops. We'll break it down in a simple way so that everyone from students to farmer can understand. Starting with recent news and developments in biofortified and bioengineered crops, uh, every student preparing for AFO and NABARD will be familiar with these two news. The first news is in August 2024, Prime Minister unveiled 109 new crop varieties developed by ICR. These varieties were designed to be climate resilient and nutritionally enriched to address the challenges posed by climate change and malnutrition. The second news is in December 2024, China approved 5 gene-edited and 12 genetically modified types of crop of soybean, cotton and corn. The aim behind the China's aim behind this was to boost their crop yields, reduce import reliance and ensure food security. So from these two news, it can be concluded that biofortified crops and bioengineered crops, or we can say genetically modified crops, are very important for examination point of view. The examiner can ask the basic details or basic concept about these two terms, the methods through which these two crops are made, what are their challenges, what are the opportunities, how can impact the human health, what are their impact on agricultural ecosystem. So in these ways, these two topics are very important from examination point of view. So I request every student watching this session to carefully and be attentive during this session, and I have compiled every topic, every point and every area from where the examiner can ask the question regarding these two topics. So I request everyone to be attentive during this session. Moving on. So moving on, the question arises, why do we need nutrient-rich crops? So the answer is malnutrition. So do you know that over 2 billion people suffer from hidden hunger? This means that they eat enough food but lack essential nutrients like iron, zinc, and vitamin A. This leads to health issues like iron deficiency causing anemia, vitamin A deficiency causing blindness, zinc deficiency causing weak immune and malnourishment. So to solve this, the scientists have developed two advanced solutions, which are biofortification and bioengineered. But what are they? How do they help? And which one is better? Let's find out all about this in today's session. There is this term malnutrition. So many will be familiar with this term. This term. Actually, malnutrition is not just about eating less food. It's also about eating food that lacks essential vitamins and minerals. This is called hidden hunger. Okay. So if it happens when people eat staple foods like rice, wheat, and maize, which do not have enough nutrients. For example, for let's say a bowl of plain white rice may fill your stomach, but it does not fully give you the required nutrients like iron or vitamin A. So these all leads to health problems like anemia, blindness, and weak immunity. So from here, I would like to put a quick question for everyone who is watching this session. Can you guess which common food is naturally rich in vitamin A? Uh, Please drop your answers in the comments below. Moving on. The first, we'll deal with the biofortified crops first. So biofortified crops or biofortification, biofortified crops are made from biofortification. So it's important to learn the process first. So biofortification is a process of naturally increasing the nutrient content of crops through conventional breeding, agronomic interventions, or advanced non-GMO breeding techniques. So unlike genetic engineering, biofortification does not involve direct gene modification or insertion of foreign DNA. So, for example, zinc-enriched wheat, iron-rich pearl millet, and orange-fleshed sweet potato. These are the examples of commonly available biofortified crops in India. Um, so, can you guess the nutrient which is most commonly increased in the biofortified crops? Like, which is it? Iron? Is it iron? Is it zinc? Or is it vitamin C? Drop your answers in the comments below. Moving on to their breeding strategies. The biofortified crops varieties require advanced plant breeding techniques that enhance nutrient content while maintaining or improving yield, pest resistance, and climate resilience. The major breeding strategies include conventional breeding, market-assisted selection, genome editing, and agronomic biofortifications. I have noted the years or time period which require these breeding techniques to be completed. Let's discuss in detail each of the breeding strategies. First is the conventional breeding. The conventional breeding involves generally crossbreeding of two parent plants, one with a, let's say, one with a higher nutrient content and another parent with a high yield or disease resistance or any other desirable parents. So they are crossbred. The steps involves generally selection of the donor parent, nutrient-rich donor parent, and high-yielding recipient parent. They are then hybridized, or we can say cross-breeded. After that, they are repeatedly back crossed to fully flourish the required high-yielding characteristics from the parents to the we can say to the offspring. Then their field testing is done and then the final release for their cultivation. The advantage can be concluded that they have non-GMO, which is genetically modified uh, organism type of approach, which is widely accepted by consumers and regulatory bodies. They are cost effective. They do not require any complex genetic engineering technology, so they are cost effective, and they are widely accepted in various crops. Just for example, it is used for rice, wheat, maize, beans, and millets. The challenges is that it is very time consuming. It requires 7 to 8, 10 years of time to develop a new variety. Depends there is also a limitation of genetic variation because it depends on naturally occurring higher nutrient varieties. The more you get the natural occurring high nutrient varieties, the more you can do selective or conventional breeding on them. So there is a limitation of the available high nutrient varieties. The example for conventional breeding is zinc enriched wheat, which is made by ICR and Harvest Plus. The second method is marker assisted selection, which is MAS. Here there is a use of molecular markers, specific DNA, which is a specific DNA sequences linked to high nutrient traits to achieve or to extract the selection in breeding programs. So in this way, the marker assisted selection is done. The advantage is that it takes 5 to 7 years instead of 10 years, which we have seen earlier in the conventional breeding. So it is faster than conventional breeding. There is also a precise selection. Precise selection in the sense they select only plants with high nutrient traits. So they are very precise in the selection. Thirdly, multiple trait combination. That means we can select at one time multiple traits, like we can select yield, nutrition, and disease resistance, all these together, all these three at one time. So this is also a very good method. The challenges, we can say that it requires advanced lab because as we can see, it uses DNA markers, the isolation of DNA, then injection of DNA. These all things require very advanced and controlled environment, and the labs of these labs conducting these selection procedures are of very initial high cost. So these all are the challenges of this strategy, or we can say method. The example here is iron-rich pearl millet in India, which is developed through this method. Third is the genome editing. So genome editing, in this genome editing, the CRISPR-Cas9, which is a precise gene editing tool that modifies the plant DNA to increase nutrient content without introducing foreign genes, unlike GMOs. So it is used to activate or silence gene involved in nutrient uptake and storage. The advantage of this genome editing is that it is faster. It requires less time than marker assisted selection as well. So it is very faster technique. We can make new varieties in 3 to 5 years, say. Then no GMO, or we can say no foreign DNA is required, so it extremely avoids the regulatory concerns whatsoever. And it is also precise and targeted because there is usage of DNA, so it is very precise and targeted. Only the required trait is selected and it is injected. So here the example of this method is enhanced iron and zinc enrich, which is actually, this method is generally performed in China and Japan for incorporating iron and zinc qualities in rice. Next is the genetic engineering, which is GM crops. In this, there is involvement of inserting genes from other organisms to improve the nutritional value of crops. Unlike conventional breeding, it allows transfer of traits between unrelated species. The advantage of this technique is that it can introduce completely new traits in with very short period of time, or we can say 5 to 7 years. It can solve severe nutrient deficiencies in vulnerable populations, so as to address the deficiency problem. The challenge is that there is very strict regulation and public concerns over this method. So in this way, genetic engineering challenges are completed. The example here is the golden rice variety, uh, which is a vitamin A enriched developed using, which is vitamin A, which is developed using genetic engineering method. Last method is the agronomic biofortification. In this method, instead of modified plant genes, nutrients are added to soil or applied directly to the crops to improve their nutrition content. Generally, the method, or we can say process, through which agronomic biofortification is achieved is soil fortification. Here we have, in soil fortification, addition of zinc or iron fertilizers are done to improve the soil nutrient uptake in crops, like zinc fertilizers in wheat fields. Then foliar spray. In the foliar spray, we spray micronutrient solutions like zinc sulfate, or iron spray on rice field to increase the iron levels. Then microbial biofortification. In this, beneficial microbes like rhizobacteria are sprayed to increase the nutrient absorption. The example could be iron fixing bacteria applied to legumes. So in this way, agronomic biofortification is done. Advantage is very quick results. Nutrient content increases in the same growing season, so the results are very quick. Cost effective for farmers because it can be used, or this method can be integrated into existing farming practices. So it is very cost effective for farmers. Challenge is that it is less stable that or it is less stable to genetic solutions because it requires continuous application. And in genetic solution, there is only one time you have to insert the gene, and final product is the desired trait is there present in the offspring. But to uh, gain the nutrient content in the field, you have to continuously apply the solution every year, every ongoing season. So it is very continuous or tedious application procedure. Then effectiveness varies by soil and climate conditions. With region to region, the effectiveness varies. Okay, then lastly, its example. The example is zinc biofortified rice developed in the India through soil zinc application. Here I have provided the summarized table of all the breeding strategies of biofortification, the time required by them, the acceptance, the cost, and the examples for the easier revisions for the students. Moving on, uh, here the biofortification, as we can guess, or we can say that the biofortification is very promising strategy to combat micronutrient deficiency, which is hidden hunger, particularly in developing countries. However, despite its benefits, several scientific, economic, social, regulatory challenges hinder its larger scale adoption. At that time, technological advancement and policy support provide opportunities for expanding biofortification research and implementation. So, in this manner, we'll discuss the challenges and opportunities of the biofortification. The challenges first is the limited genetic variation. Many staple crops have low natural genetic variability for certain micronutrients, making it difficult to develop high nutrient varieties through conventional breeding. So, for example, we can say rice, which naturally has very low iron and zinc levels, has limited potential of traditional breeding approach. So, in this way, there is limited genetic variation among the crops. Second is uh, trade nutrient trade-off, which means that with the increasing nutrient content in crops can sometimes have a negative impact on yield and productivity. For instance, higher iron or zinc levels in wheat and rice may lead to changes in metabolism, reducing overall grain size or weight. Okay. The next is bioavailability issues. In this, we can say that not all nutrients in biofortified crops are fully absorbed by the human body, because some crops may contain anti-nutritional factors like phytic acid, phytase, or oxalic acids. These are all anti-nutritional content found in the crops that inhibit iron, zinc, or calcium absorption in overall. For example, iron in millets may be poorly absorbed due to the presence of tannins and phytates. So it could be a major challenge for biofortified crops. Fourth is climate change impact. In this, we can say that the rising CO2 levels due to climate change can reduce the mineral and vitamin content of the crops. There are studies that shows that the crop grown under high CO2 conditions have lower zinc, iron, and protein content. Okay. Next is slow farmer adoption. Farmers often prefer high-yielding or disease-resistant varieties over biofortified crops if they do not provide a direct economic benefit. So, in this way, there is very uh, limited adoption by the farmer. Next is consumer acceptance. Many consumers are aware of biofortified crops or hesitate to adopt them due to color, taste, or unflammability. For example, when the golden rice was introduced, it has yellow color, which may not be preferred by which is still now is not preferred by consumer due to because we have seen every time we have seen white rice, so it is very uh, unaccepted by consumers due to the yellow color of that particular rice. Okay. So next is regulatory barriers. GMO crops regulations, we can say they have very delay approval of genetically engineered biofortified crops such as golden rice and high zinc rice were one of the prominent examples. So strict biosafety laws in some countries prevent farmers from adopting genetically modified crops. So for this, we can strengthen the scientific basics for the regulatory approval, so we can encourage the use of non-GMO biofortification techniques such as conventional breeding and mass, which is marked assisted selection technique. Last is the high R&D, which is research and development cost. Developing biofortified crops require years of research, extensive field trials, and nutritional validation studies. So to, we can say, for the solution of this challenge, we can say that government and private sector should provide fund for biofortification programs, international collaborations like we have seen earlier, Harvest Plus, ICR, CIMET, to share research resources, which would help the entrepreneurs to do better research and development regarding the biofortification. Moving on, the opportunities we can, what are the opportunities we can achieve from biofortification? First is the biotechnology and CRISPR advancement. We can say that the CRISPR-Cas9 and gene editing allows precise modification of genes to increase the nutrient levels in crops without introducing foreign DNA. So there is a future scope of developing nutritionally superior crop faster with greater precision and with less time. Next is agronomic biofortification. In this, we can see a scope that there will be a larger scale adoption of nutrient-rich fertilizer in staple crop production, crop producing states or staple crop production methods. Next is government policies and supports. In this, government should help in expanding the, or government should step in providing back-end seed distribution programs and farmers incentives so as to support the biofortification research. Next is public-private partnership. In public-private partnership, more investment in research development and large-scale implementation should be done, because the collaboration between research institutes, private seed companies, and government will help in accelerating the adoption of biofortified crops. Next is growing consumer awareness. In consumer awareness, expanding biofortified crops in local and global markets will can be increased through using like media coverage and education campaigns to adopt, so as to help consumer to adopt nutrient-rich foods. Next is global research collaboration. In global research collaboration, like organization of FAO, WHO, CGIAR, ICAR, these all organizations can help in emphasizing the biofortification as a key tool for achieving a sustainable development goal. There is also a sustainable development goal 2 for zero hunger, and which would help in decreasing the malnourishment problem found in developing countries. So in this way, we have covered the major points or major areas of the biofortification or biofortified crop. Next, we'll deal with bioengineered crops. Okay, so bioengineered crops are also called as genetically modified crops, or GMO crops, are developed directly altering the plant DNAs to introduce a new trait, including higher nutrient content. This often involves insertion of genes from other organisms. Okay, so I would like to put a quick question for all the students watching this session. What do you think the first commercially grown genetically modified crop was? What do you think what was the first commercially grown genetically modified crop was? Drop your answers in the comments below, and we'll see how many students know about this answer. Okay, so after knowing what is the genetically modified crop or bioengineered crop, we can, here I have presented all the methods of genetic modifications done in the crop. First is the recombinant DNA technology, which is also known by transgenic approach. Here, it is the most common method of genetic modification for developing a bioengineered crop, where a gene from one organism is inserted into another organism's DNA to introduce a new or desirable trait. So in this, the steps are very common for any genetic modification process, like identification of desired gene, then isolating the targeted gene using restriction enzymes, inserting it into a vector like plasmid, then transfer into plant cells through using of various method like agrobacterium method, gene chem method. In this way, Then next is selecting or modifying the cell culture in laboratory and screening the successful gene insertion. And lastly, the field trials before making them for commercial release. Okay, so example of the recombinant technology, prominent examples would be golden rice vitamin, which was modified for having vitamin A production, then BT cotton, BT brinjal. Okay, the next is next method of genetic modification for bioengineered crop is CRISPR-Cas9 gene editing, which is a non-transgenic approach. In this, a precision, as I have said earlier, CRISPR-Cas9 is a precision genome editing tool that allows scientists to make targeted modification in plant DNA without inserting foreign genes. Unlike traditional GMOs, CRISPR-Cas9 does not introduce external DNA, making it more acceptable technology in some regions. So here the steps are also similar, like first we target the DNA selection for or particular DNA sequence which needs to be modified. Then we'll guide the CRISPR-Cas9 to identify the specific location in the genome. Then Cas9 helps in the action of cutting the DNA at a desired location. And finally, the DNA repair and modification process concludes the genome editing procedure by either disabling a faulty gene or replacing it with beneficial mutation. Okay, so example of this method is high yielding tomatoes which were edited for faster growth and improved fruit size, low gluten wheat which was very beneficial for people with gluten tolerance. Then third is drought tolerant rice which was edited for resilient in water scarce region. Okay. The third technology used for genetic modification is RNA interference technology. In this method, there is a silencing of specific genes in plant to reduce or eliminate unwanted traits without adding foreign DNA. Okay, so in this technology, the step is firstly designing the RNA sequence. Okay, to create a small RNA molecules that match the target gene. In this way, the RNA sequences are created. Then the RNA molecules are introduced in plant cells where they bind to messenger RNA of the target gene. Then finally gene silencing or gene expression, where the plant cellular machinery destroys the mRNA, preventing the production of unwanted protein. In this way, the RNA interference technology concludes. The example of this method is Arctic apple, which was a non-browning apple, was developed by using RNA interference technology. Next is innate potato, which was a potato with low acrylamide content, which was modified to reduce the chemical which is harmful during frying. Okay. Next is gene gun method, which is very prominent method for genetic modification. In this gene gun or ballistic method, gene gun, which is also known as ballistic method, is used to directly shoot the foreign DNA into the plants under high pressure. Okay, so there is also alternate method for this, which is agrobacterium mediated transformation, which is especially useful in crops that are resistant to bacterial transformation. In that particular type of crop, agrobacterium mediated transformations are generally used. Okay, so the steps for this method is that first there is a coating of microscopic particles with DNA is done. Gold or tungsten nanoparticles are generally considered for this step. Then there is high pressure bombardment of the plant cell using high pressure gas is done. Third last is the DNA integration. The DNA is then integrated into plant's genome, resulting in a modified plant. And lastly, plant regeneration. There is a selection and regeneration process is done to conclude this method. And the then developing into a entirely new plant. Okay. So example of this method is herbicide-resistant maize and soybean, which were developed using the gene gain method, and transgenic rice, uh, where there was introduction of genes for pest resistance and improved grain quality. Okay. Then final method of the genetic modification for bioengineered crop is agrobacterium mediated gene transfer, which is most common among all the methods which I have discussed earlier for bioengineered crops. Here the agrobacterium tumefaciens is a naturally occurring soil bacterium that can transfer its DNA to plant cells. Okay, so scientists use this property to deliver desired genes into plant. So the if we want to discuss the steps, the first step is that we use the gene insertion into firstly we gene insertion technique into a agrobacterium, which means scientists introduce desired gene firstly into the agrobacterium plasmid. Then the modified agrobacterium infects plant cells and transfer the desired gene. Then there is the integration of gene into the plant's genome, and they start expressing the desired plant trait. And then final successfully plant cells are then grown into whole plants. Okay. So the example of the agrobacterium mediated modified crops are BT cotton and virus resistant papaya, which is known by rainbow papaya. Okay. Uh, me- I mean, the rainbow papaya actually is resistant to ring spot virus. Okay. So in this way, the method of genetic modification in bioengineering crop is completed. I want to put a question here for students to answer them in the comments below. Which was the first, or sorry, which genetically modified crop is most commonly grown in India? Whether it is BT cotton, golden rice, or GM crop. Okay, write your answers in the comments below. If any students who have watched earlier session of GMO crops would be able to answer this question very efficiently. So let's discuss about the advantage and disadvantages of the bioengineered crops. So it can be seen that higher nutrient enhancement can be achieved because it allows precise nutrient control beyond natural breeding limits. So we can achieve higher nutrient enhancement through these techniques of, or we can say, bioengineered crops. Next is introduction of novel traits, like scientists can introduce entirely new nutritional benefits. Okay. The next is a faster process. In this, genetic modification is much quicker than traditional breeding. So it holds major importance, or we can say major advantage for bioengineered crops. The disadvantage can be said that first is health and environmental concerns, like some people fear unintended effects of genetic modification. So they are generally not willing to accept bioengineered crops. Next is regulatory hurdles and public resistance. In this, many countries like strict have many countries or many developing countries have strict GMO regulations due to its safety concerns. So it is also a major disadvantage for bioengineered crops. Lastly, cost and technology requirements, like genetic modification requires advanced laboratory and is very expensive to develop. So it is also a major disadvantage for bioengineered crops. Finally, I have summarized all the areas of biofortified crops and bioengineered crops in this table to for a faster revision for the students. They can see the table after um, completing the session. Now that now that many students know the differences which of both the crops, I would like to know what would you think, or what do you think is better for the future of food security? Is it biofortification or bioengineering? I would like to know your opinions in the comments below. Uh, After completing this session, I'll be posting few um, great questions on both biofortification and bioengineering crops in our Telegram channel. So please, I request every student watching this session to join our Telegram channel by Agri Clarity to attempt those quiz in following days. Finally, I would like to conclude the session with that is both biofortification and bioengineering are very essential for fighting malnutrition. I feel so. Biofortification is generally more natural and widely accepted, but GM crops provide faster solutions. So I think government and scientists are working together to make more uh, nutritious and sustainable food. So please drop your suggestion in the, or your opinion in the comment below. Which do you think is the better solution? And don't forget to like, share, and subscribe to Agri Clarity for more such informative content. We have also launched a test series for students preparing for AFO. Please check the details of the scores given in the description below. Thank you.